Okay, welcome back. So, what we're going to be talking about now are all the different organelles. So, we're talking about eukaryotic cells at this point. So, I'm going to go through each of them and kind of give you the major players and what they do. Some of the pictures might have shown some more different organelles, but we're going to focus on the big guys and have the major roles. The first one I want to talk about is the nucleus. He's pretty much the boss of the cell if you want to think about it. Now, this organelle has two major functions. First off, it's where we're going to store your hereditary material or your DNA. So all the information is going to be stored in this nucleus. This entire picture is the nucleus except for these little foldings right here. Now, within the nucleus, you're going to see an inner part too. This is going to be called the nucleolus, this very center part, like the core of the earth, if you want to think of it that way. Now, in the nucleolus, this is where we're going to make the ribosomes. The DNA is going to be located in this outer part in your linear chromosomes. So we store information as DNA, and it's also going to coordinate all the cell's activities. So it's the boss. When we need to grow, we need to make more proteins. We need to prepare for cell division. We're getting ready to reproduce. Pretty much he's in charge. It's like City Hall. The blueprints are your DNA, but when something needs to happen, the mayor, if you want to call it this way, he tells us what to do and why. Now, within your nucleus, we organize our DNA into what we call chromatin. Basically, this is DNA plus protein. So within your nucleus, regardless of whatever type of cell it is, any cell in your body, you took out all the DNA and laid it out end to end, it'd be 6 feet long, which I find that fascinating. From one individual cell in your body, there's 6 feet of DNA. So if you took all the cells in your body and laid out all the DNA, I think you go to the sun and back 3 times or something like that. Really fascinating. But to organize all this DNA and fit it into the nucleus, we wrap it around these proteins called histones, and it looks like a little beaded necklace right here. So the term is chromatin is your DNA plus proteins to help organize it. Now, the nucleus is surrounded by a nuclear membrane, which is made up of phospholipids, which we'll talk about in a little bit more detail later. And it's going to have what we call nuclear pores. Now, the pores are important because we have to get stuff in and out of the nucleus. When it comes to regulating your DNA, it's done by proteins, and they have to come in and out. When we go through transcription, we're going to make a copy of the DNA, and we just send it to the ribosome, which is located out of the cell, so it has to leave this way. So the nuclear pores allow us to transport things in and out. So nucleus, super important, probably one of my favorites because it's the boss and it's where all the DNA is located. Has several functions. There's that the nucleolus, it's where ribosomes are made, but that kind of gives you the idea why he's super important. Now, I've introduced the plasma membrane, and I'll go in more detail in the next lecture on it, but the plasma membrane is going to be your exterior of the cell. It's going to be made up of that phospholipid bilayer. So if you remember, there's those hydrophobic tails that are going to face inside, and then you're going to have those hydrophilic heads that love to be outside. So your cell is mostly water. You're going to have water on the outside and the inside of your cells. With this phospholipid bilayer, it's going to make this barrier where there's going to be a no water zone. So it makes this outer layer, if you want to think of it as the city limits. It's the outer boundary of your cell. It also makes up the outer membrane of your organelles. So that nucleus that we were just talking about, it is surrounded by a nuclear membrane. It's made up of these phospholipids. Now I'm going to talk about this in a little bit more detail, but I want you to understand the plasma membrane. I'll use this term fluid mosaic model to talk about it. The reason I do that, I want you to appreciate that the plasma membrane, it's not static. It's kind of loosey-goosey. It's flowing. Things are moving around. It's not rigid. And it's also got different components to it. So we talk about the phospholipids, but there's also cholesterol. There's proteins that will act as channels. We have glycoproteins and glycolipids that are used for cell connection and cell communication. There's a lot going on here. If you just look at this picture right here, you see a lot of things happening. Another thing, it's semi-permeable. So it's going to regulate what comes in and out. So think of it as like the gatekeeper, it's like this outer fence of your cell, and it's going to regulate who can cross, how they can cross, and what leaves. It does serve as protection, so it keeps everything from the inside from getting to the outside. Now, depending if what type of cell you are, you might have a cell wall. Now, the cell wall is going to be located right next to the plasma membrane, so plants fall in this category. Some fungi and bacteria and algae fall in here too. I wrote bacteria twice, I just saw that. I'll have to fix that later. But it's going to be surrounding right next to the plasma membrane. Now, the cell wall has several functions. It's going to provide protection, because think of plants, they can't get up and run away from a predator. And it also provides structural support and structure and support. If we remember, we talked about cellulose and how it makes the rigidity of the cell wall and provides structure and support for the plants. It all comes down to this outer part right here. Next up is the Endoplasmic Reticulum, and we call it the ER for short because that's like a long word with lots of little different syllables. Now, this is a network, and it's going to be a single layer membrane that's continuous from the nuclear membrane. So here's our nucleus right here. And remember, we had that nuclear membrane that surrounded it. What the ER is, it's like an extension of the nuclear membrane, and it's all these different folds. So it just keeps going and folding and folding and folding, providing lots of surface areas. Now, there's two different types of ER, and it all comes down to the presence of ribosomes. Now, if you have ribosomes present, we're going to call it the rough ER. So think of them as like little studded little beads that are on the outside. So if you rubbed your hand on it, it'd have a rough appearance. If you do not have ribosomes, it's called the smooth ER. Now, the presence of the ribosomes helps determine what the function is going to be. So the smooth ER, so no ribosomes, it's going to be important for making your fats, your lipids, also for making your hormones, and also breaking down some of your toxins. The rough ER is going to have ribosomes, and it's going to be important for making membrane-embedded and secretory proteins. So mostly protein work right here, fat work right here. One looks very similar to the ER, but it's going to be on its own location, is called the Golgi apparatus. Now, this is a set of flattened membrane sacs. It looks like if you took a bunch of balloons and flattened them out, it's what it is. Now, what this does is he has like, I kind of call him like the post office. He kind of gets things ready for packaging. So once a protein is made, either by the rough ER or by a ribosome, it'll be transported in a vesicle, which is like a little shipping container, and taken to the Golgi. It's going to fuse with Golgi in what we call the cis phase, and then as it goes through this, it's going to go through a series of modifications. It's going to help that protein fold. So remember, we talked about protein structure. There's primary, secondary, tertiary, and then quaternary that some get to. What's going to happen in the Golgi, it's going to take that protein from primary and start doing that folding and twisting and getting into its final shape. Once it does that, it's getting ready, gets to what's called the trans phase. And what happens is we then package it into another vesicle, these little round guys right here, and we put like a chemical tag on it, like an address. And then we're going to exit, and that chemical tag is going to direct exactly where it needs to go in the cell. So maybe it's a protein that needs to leave the cell, then it'll be taken to the plasma membrane and taken out. Or maybe it needs to go to the nucleus. So he does a lot of the modifications, like the UPS or a post office. You take something and they'll package it up for you, fold it up, put the address on it, and get it prepared and ship it off. Now, vesicles, I just mentioned briefly, I talked about them, but these are just little small membrane sacs, and we use them to move things throughout the cell. We can move them in the cell, out of the cell, throughout the cell. They're very, very helpful. So we can use them to transport between the different organelles, like I just talked about, moving from the rough ER protein that was made, moving it to the Golgi, and then the Golgi moving it to maybe the plasma membrane. Now, we can also use it to move things in and out of the cell. So example right here is we're using a vesicle to move a food source. So here's our cell right here, and here's a little bacterium. Well, he engulfed him, and we'll talk about phagocytosis in a few slides, but he kind of was hungry. This is a food source, so he circles him up, and he puts him into a vesicle. Now it's like a little box, he's going to boxed up his dinner. Now that's going to be taken, and it's going to fuse with another type of vesicle that's going to go through and do the digestion of it. So these transport vesicles have several purposes, moving stuff around in and out of the cell. One that's not shown in this diagram is called a peroxisome. It just shows up as a little ball on most cell diagrams. But it's going to be important for helping eliminate waste by removing hydrogen to produce hydrogen peroxide, and then your cell later converts that to water, because we got to get the waste out of your cell. One that is shown right here is called the lysosome. I call it your portable stomach. And what they're going to do is they're going to digest the particles that your cell brings in. So this little bacterium, what's going to happen is this vesicle is going to fuse with the lysosome. So these are made up of those phospholipids, so that's why they're easily able to fuse together. And then it's going to have a lot of digestive enzymes in this lysosome to break this down into smaller components that can then be fed into maybe cell respiration, or smaller components that we can then use for protein synthesis, or all the kinds of things in the cell. Now, if you're a plant cell, you're going to what we call a vacuole. Think of it as like a storage locker, large storage center in plant. So you'll see a large central vacuole in this little cartoon image right here. Now, in the central vacuole, they'll store water, sometimes some compounds for herbivores, enzymes in there. If you are a plant grower, and plants, they don't know when their next water source is going to be. We're in summer right now, so I try to water my plant outside very often. My hibiscus, I looked at it yesterday, and when a plant needs water, it gets that wilty appearance and everything kind of shrivels and it kind of looks kind of sad. Well, what's happened is it's used up its water reserves in that central vacuole, and as it uses up that central vacuole kind of shrinks in on itself, and then it kind of pulls away from the cell wall, and the cell kind of looks kind of shriveled. Now, when you water it, what happens is the water is replenished, and it's going to store it in that central vacuole because it doesn't know when the next drink's coming, and your plant kind of perks back up. All right, good old mitochondria, the powerhouse of the cell. Now, this is going to be the site of cellular respiration, which we'll talk about in the next unit. But basically, that's where we're going to take your carbs, your sugars that you might have just ate, and we're going to use them to make ATP. Now, the mitochondria is quite cool. It's a double membrane organelle. So it has both an inner and an outer membrane. And I'll talk about why it got that in a few slides. What's also really cool, it has its own genome. So there is mitochondria DNA throughout mitochondria. It's separate from the nuclear DNA, which is really cool. Now, you might have heard the term, I love to watch all the crime shows. That's just me. What I love to do. I can watch Law and Orders and all that kind of stuff all the time. You might have heard the term mitochondria DNA. Now, it was thought that all the mitochondrial DNA only came from a maternal source. It was thought that when the egg and sperm come together, pretty much the sperm just drops off as DNA, and the egg had all the organelles to start off the zygote. But actually, since we're getting more and more technology, we're able to sequence stuff like crazy, they started sequencing the DNA in the mitochondria, and they found both a maternal and possibly paternal mitochondria DNA. I can guarantee in the textbook it says it's only maternal, but this is a cool new thing in the field, and it really does emphasize that with science, we're always learning. Yeah, we have our facts, but as we get more technology, we learn new things, sometimes those facts have to be altered a little bit. So we're not wrong that there's maternal DNA, but there might be some paternal. One of the things that the crime shows love to do is link maternal DNA, or maternal mitochondrial DNA, and if this paternal thing, really catches on, which is more and more people starting to do it. There's a debate about the technology and methods going used for it, but could rewrite a few books. I think that's kind of cool. How it came out to be an organelle, it's this process called endosymbiosis, and I'll talk about it in 2 slides. Next slide. All right, chloroplast, the cousin of the mitochondria. So the chloroplast is where photosynthesis is going to happen. It is a double membrane organelle just like the mitochondria, so it's going to have this inner and this outer membrane. It's also going to have these stacks that look like stacks of coins. These are going to be your thylakoid membranes. They're going to provide a lot of surface area and is one of where the light reactions are going to happen for photosynthesis. And it's also where we're going to have this green pigment called chlorophyll. So it's a photosynthetic pigment. It's going to be important for triggering the light reactions. Now, what's really cool about the chloroplast, because of that pigment chlorophyll, this is the only organelle you can see without staining a cell. If you were just to like take some cells and put it on a microscope slide, you probably wouldn't see much because in order for us to actually see a lot of the organelles or different parts of the cell, we have to use stains. And with the chloroplast, since it has that natural pigment color, that green, you don't have to go through the process of staining cells. You can actually see them. So all these little green balls that you see, each of those are an individual chloroplast. I think they're kind of cool. So how did the mitochondria and the chloroplast come to be there? Now they are, remember, eukaryotic organelles, and it's thought that they came across this theory called the endosymbiotic theory. I call it tasty snacks. Now remember, both the mitochondria and the chloroplast have their own DNA. So that's kind of indicating maybe at one point in time they were their own different organism that was engulfed by a different one, and instead of breaking it down for food, it decided to keep it around and give it a job. So I'll kind of walk you through this diagram right here. So we have our very primitive cell. He's starting to get more and more complex. So right now he's in here, but he's hungry. I gotta eat. I'm super hungry. It's about lunchtime for me too. So this photobacterium is just kind of minding his own business, and he's engulfed by the blue cell and taken in. Now, instead of breaking him down for smaller parts and send him to the lysosome to break him down in small parts and use them for food source and everything else, he's like, hey, this guy is really efficient at making energy, and I'm not that good at it. So he kind of kept him around and gave him a job, and that's how the mitochondria is thought to be there. Same thing with plant cells. Now, with the chloroplast, it's thought to be a cyanobacterium, so it's a little bacterium that can do photosynthesis. And, he's hungry, looking around, nom, nom, nom. See that engulfs it, and he's like, hey, that's really cool. You can make food from sunlight. I can't do that. So instead of breaking it down, gives it a job, calls it the chloroplast. Now, what's really cool about plant cells, they do have both mitochondria and chloroplast, but we don't have chloroplast. I've had one student ask, could we? But then we'd have that green pigment, so I don't know if you'd want to be green, but it'd be kind of cool to make our food from sunlight, but, I do love a good cheeseburger now and then too. All right, just a few more. Next up is the ribosome. It's the site of protein synthesis, and we'll talk about him a lot when it comes to translation. It's going to be two subunits. It's going to have this bottom subunit and a top unit, and this is where we're going to make your proteins, and we're going to use this messenger RNA molecule that we're going to make in a process called transcription, and we're going to use that to build that chain of amino acids that will be folded up and made into a protein. Now, these ribosomes, some of them can be studied on ER, which makes up that rough ER, and a lot of them are just free-floating in the cytoplasm. So you might have heard these terms cytoplasm and cytosol, and they can get kind of confusing. I think people get them mixed up most of the time. You've probably heard cytoplasm more. Now, cytosol is a cellular fluid. It's mostly going to be water, have some proteins and salts in it. But the term cytoplasm is actually all that fluid between all the different organelles plus the organelles. So I use this analogy to kind of help it explain to my students, and some of them says it really does help. So I'm Polish, you know, I was born and raised, and one of the things I love little grannies love to do is to make Jello salad. If you've never had Jello salad, it's Jello with a bunch of fruit chunks in it. I just love it. You can't go to a church picnic and not have 18 of them. Sometimes they look kind of scary. This one looks scary. So think of Jello like the cytosol, the fluid that's going to be between all the different parts organelles. If you want to think of the fruit chunks, which I don't know what kind of fruit that is, maybe some pomegranate, I'm not sure. Think of those as the organelles. So the Jello salad, both the Jello plus the fruit chunks, is like the cytoplasm. So the cytoplasm is both cytosol, so all the fluid between it, plus the cellular organelles, so minus the nucleus. Now, I want to introduce like cytoskeleton a little bit. So we need to have some building, some framework inside the cell, and that's going to be our cytoskeletal elements. Think of it as like the scaffolding, you know, your framework for your cell. Now, this is made up of proteins, and it's going to help organize different functions within the cell and different activities. It can provide like highways to move proteins throughout the cell. And what's really cool, some of these can be easily broken down and reorganized as needed. So it could be like temporary scaffolding that you might need depending on what we're doing. There's three main types. The first one's intermediate filaments. Right, on this top image right here, and kind of think it's like a mesh surrounding the interior of the cell membrane and the nuclear membrane. If you ever done the little art and crafts projects where you take the string and you put it in glue and wrap it all around a balloon and kind of pop it, think of that kind of like that mesh thing like around the balloon. That's kind of like your intermediate filaments. Now the microfilaments, they help maintain cell shape and they're very essential for movement. In this bottom image here, they're mostly going to be on the exterior and they can be broken down and reassembled quite easily. One little guy I like to show a video of is a guy Amoeba. He's a little protist, and he kind of, it's like that blob. You can just looks like a snot booger just moving around, and it has to use his foot called a cytopodia, which is a false foot, to move. And what's happening is as he's moving, he's breaking down and building these microfilaments, and you keep changing his shape. It's really cool. And finally, the microtubules. Think of them as like cellular bones, and they're going to be very essential for cell division because you're going to see when we get to a point where we're ready to split our sister chromosomes to opposite sides of the cells, we're going to use microtubules to build them, and then we're going to attach them and then pull them to the opposite poles. And then when we're done, we're going to break them apart because we don't need them at that point. So last thing I want to really introduce new stuff is some cell junctions. And I say animal / plant, but I put the last one right here. Now, what's going to happen is sometimes we got to keep cells together. And depending on what type of tissue it is, it might determine how the cells need to be attached to each other. Think about the linings of your stomach and your intestine. You might want to make sure you keep all those stomach acids just in your stomach. You might want your cells to have these tight junctions. When that happens, they're going to be super close together and you're not going to be able to get anything in between them. Like they're glued together. It's going to make a watertight connection. You could not get through them. Now a desmosome, boom, boom, boom. So these cells are kept together. It's like if you staple two pieces of paper together. So you can move them around a little bit, but they're still connected. These are then those intermediate filaments that we talked about on the previous slide. What they do is they can actually go between these little holes right here, and they can be shared. So it keeps them together, but it will allow like fluid to flow through. So we keep them together, but they're not super tight. Next one are your gap junctions. Now, your gap junctions are going to be channels, all right? And what that allows us is to share chemicals and water and all kinds of molecules between two different types of cells. Like I said, sharing's caring. And finally, the plasmodesmata. These are very similar to gap junctions, but what they do is they allow sharing between the plant cells right here. So the last slide here has just fun facts, which I think is kind of cool. You're not required to memorize these, but if you want use trivia to share with your friends, that's here for you, which I think is kind of cool, you know, and kind of gross at the same time. So I'm going to stop this one and we'll move on to the next lecture.